Very good morning to everyone. Welcome back to the session 2 of this chapter called as Biotechnology Principle and the Process. So hope you remember in the last class we have studied about what biotechnology. What is biotechnology? What is biotechnology? Biotechnology is the use of cells or the tissue or the organs or the organism to produce a product which are useful to the human mankind, right? So we also studied about the techniques that are used in the process of biotechnology, right? So all this we have studied. Now let us move on to the further concepts of this very interesting chapter called as Biotechnology Principle and the Process. So we are going to study about a very important concept and also a very important word that is called as what? Plasmid. Plasmid. So what are this plasmid? Usually we know that the plasmids and the bacteriophages are commonly used as what? Vectors. So this plasmid and the bacteriophage. So all these are used as what? Vectors. What? Vectors, right? That is the plasmid and the bacteriophage. So what do you mean by bacteriophage? The virus which infect the bacterial cells, which is called as what? Bacteriophages. Usually the plasmids and the bacteriophages are used as what? Vectors, right? Now let us understand what is this plasmids, right? So these plasmids are, so these are the anatomically replicating circular extra chromosomal DNA present in the cytoplasm of the bacterial cell. So this is a very important word which is usually asked in the examination. The term, what is the term called as what? Plasmids. So what are plasmids? The plasmids are the what? These are the anatomically, that is self. So these are the anatomically replicating circular extra chromosomal DNA present in the cytoplasm of bacterial cell, right? So you can see here, this is the diagram. So this is what? This is a circular DNA. So these are what? Plasmids. So this is what? This is the what? Bacterial DNA. You can see here, this is the plasmid. Plasmids are circular and self-replicating, that is replicating circular extra chromosomal DNA which is present in the cytoplasm of the bacterial cell, right? So this is nothing but what? Plasmids. You can see here, this is how the plasmids look like, that is circular, that is, as I told, autonomously self-replicating circular extra chromosomal DNA which is present in the what? In the bacterial cell. Now moving on to the next is, hope you got to understand what is this plasmid. Now moving on to the, what are the tools which is used in the recombinant DNA technology. The tools that is used in the recombinant DNA technology. So what are all the tools that we are used in the recombinant DNA technology? So that is, as I told, so there are, so which is used in the, what are all the things that are used in the recombinant DNA technology? That is, first one is restriction enzyme. Second one is polymerase enzyme. Ligase. Fourth is vectors. Fifth is what is fifth one? Host cells. and sixth one is bioreactor. So what are the tools used in the recombinant DNA technology? That is restriction enzyme, polymerase enzyme, ligase, vectors, host cells and the bioreactor. So it is a very important concept which is usually asked in your exam. So what are the tools that are used in the recombinant DNA technology? That is restriction enzyme, polymerase enzyme, ligases, vectors, host cells and the bioreactor. So this restriction enzymes, namely there are of two, that is exonuclease and the endonuclease, and the polymerase enzyme, and this ligases are usually called as the molecular stitches. So these are called as what? Ligases are called as molecular stitches, whereas the restriction enzymes, they are called as molecular scissor, which will cut the DNA at its specific sites. Understood? So the restriction enzymes are called as molecular scissors, whereas the ligases are called as molecular stitches. Right? You got to understand what restriction enzymes are called as molecular scissors and the ligases are called as molecular stitches. Now let us understand what is this restriction enzyme. Now let us understand what are all this by one, right? So what is this restriction enzyme? So as you know that it is a this restriction enzyme, it is a what? It is very important tool for the recombinant DNA technology. So what are this restriction enzyme? So you can see, and while reading this on your textbook, please refer the text page number 196 figure 11.1 for the restriction enzyme. You have that particular picture on that diagram, right? So these are the actually this restriction enzymes, they belongs to the class enzyme called as nucleases. So this restriction enzyme, they belongs to the class called nucleases. So that belongs to the class enzyme of what? Nucleases. So this restriction enzymes belongs to the class of enzyme called as nucleases. So these are the larger class enzyme which will cut the DNA at a specific sites. So what is the very important function of this restricted enzyme? So these are the large class of enzyme which cut, that means these are the large class of enzyme belong to the class nucleases which will cut the DNA at a specific sites. So there are of two types of restriction enzyme, that is restriction exonuclease, restriction endonucleases. Understood? There are of two types of restriction enzyme, that is restriction endonuclease and restriction exonucleases. So this restriction endonucleases are called as what? They are called as what? Molecular scissors. They are called as molecular scissors. Right? They also, we can write in a short form REN. Right? So now let us understand what is the difference between this restriction and exonuclease and the restriction endonuclease. So what is this restriction exonuclease? What is this restriction exonuclease? So these are the nucleus used to remove the nucleotide from the ends of the DNA. So what will happen here in the restriction exonuclease? These are the nucleus which is used to remove the nucleotide from the ends of the DNA. So you know that particularly the process of biotechnology, we can manipulate the genes. That means take the genes you have desired and they will join it together to your desired character. So that is what is being done in the field of what? Biotechnology, right? So it will help the restriction enzyme like restriction exonuclease, which are the nucleus used to remove the nucleotide from the ends of the DNA, right? So what are the restriction enzymes? these are the larger class of the enzyme belong to the nucleus which will cut the DNA at a specific sites, which is of two types, that is restriction exonucleus, restriction endonucleus. So this restriction exonucleus are the nucleus which is used to remove the nucleotide from the ends of the DNA. So that is, the nucleotide has been removed from the ends of the DNA. Now let us understand what do you mean by restriction endonucleus. As I told it, we can write in a short form, that is REN. So these are the nucleases which is used to cut the specific position within the what? DNA. So it is used to cut in the specific position within the DNA. Within the DNA, right? So the restriction endonucleus, as I said, these are the molecular scissors. So these are the nucleus which is used to cut the specific position within the DNA. So hope you got to know what are the difference between this restriction exonucleus which will remove the nucleate from the end of the DNA at the specific sites, whereas the restriction endonucleus, what they will do, they use to cut at the specific position within the DNA, right? So this is the difference between the restriction endonucleus and the restriction exonucleus, right? So this restriction endonucleus are the cleaving enzyme which will produced by the bacteria to protect themselves from the foreign DNA. So this restriction endonucleus are the cleaving enzyme which will produce by what? Produced by the bacteria to protect. Why it is protect? To protect themselves from the foreign DNA, that is like viral. So the virus, that is a foreign DNA, it is a virus. So this endonucleus are the cleaving enzyme which will produce by the bacteria to protect themselves from the foreign DNA. Nucleus, as I told, these are the molecular scissors. Why they are called as molecular scissors? Because they cut the DNA at a specific region. So the specific region, that region we call as restriction site. That specific position that we call as what? Restriction sites. So this endonucleus which will cut the DNA at the specific region called as restriction sites. Hence they are called as molecular scissors. As I said, restriction endonucleus or REN are referred to as a molecular scissors. Why they are called molecular scissors? Because they cut the DNA at a specific region called as restriction sites. And this restriction endonucleus was discovered by a scientist called as Hamilton Smith. So this restriction enzyme was discovered by a scientist called as Hamilton Smith. So the person who discovered the endonucleus is the what? Hamilton Smith, and he discovered, as I told, he discovered the endonucleus, and Haemophilus influenza bacteria endonucleus are also discovered. That is Haemophilia influenza bacterial endonucleus is that REN, which is also discovered by Hamilton Smith. Now moving on to the palindromes. Moving on to the next important concept, that is called as palindrome. Hope you have heard about the word called palindromes, right? So what is this? What are palindromes? So these are the groups of letters. So these are the groups of letter. So these are the groups of letter that form the same words when we read from both forward and the backward direction. So what are palindromes? These are the groups of letters that form the same words when we read both forward and the backward. For example, if you take Malayalam, so you can read it from like right to left, left to right. So it will remained as the same words, right? So that is called as palindromes. You can give another example like madam, gadag, mom, ma'am. So like that, if you read it, so these are the group of letters. When you read it from right to left or left to right, it is the same. So just like Malayalam, Gadag, Mom and like that and all. So as I told, the forward to backward or backward to forward also the same. So that is sequence that we call as what? Palindromes. Right. So these are the groups of letters that form the same words when read both forward and the backward direction. For example, Malayalam, madam, gadag, mom, ma'am. So such words are called as what? Palindrome. So what is the palindromic sequence in DNA then? So, what are these palindromes? How did these palindromes are played in a sequence in a DNA, right? So, what is this palindromic sequence in DNA then? So, it is a sequence of base pairs that reads the same on the two strand both in forward and the backward direction. So, as I told, the base pairs, that is adenine, guanine, thymine, cytosine. So, all read it the same on the two strand, that is like you can see 5 strand, 3 strand, 3 strand to 5 strand. It will same. So these are the sequence of the base pair that reads the same on the two strands, that is 5 to 3, 3 to 5. It will reach the same strand both in the forward and the backward direction. So as I told, the base pairs like adenine, guanine, thymine, cytosine that reads the same on the two strand both in the forward and the backward direction. I think you will get to understand about this when we come across with the example. So here you can see that so like a 5 strand. So if I am taking GAATC 3 strand, so 3 strand when it is written in form CTAAG, that is 5 strand. You can see here, you can read it from GAATC. Here that C has been written, right, like forward to backward. So CTAAG. So this is called as palindromic sequence in a DNA. So this is the example of the restriction site of E. coli restriction enzyme, right? That is E. coli. So this is the example of the restriction site of E. coli enzyme. Then the action of the restriction enzyme in the formation of recombinant DNA. So what is the role played by the restriction enzyme in the process of formation of rDNA? rDNA is what? Recombinant DNA. So the restriction enzyme cut the strand of DNA a little away from the center of the palindromic sites. So what will the RNA do? That is REN do. So the restriction enzyme will cut the strand of a DNA at a little away from the center of the palindrome sites, but between the same two bases on the opposite strand. So this leave what? So this leave a single stranded portion at the ends. So there are overhanging which will stretches called as sticky ends on each strand, and these are named so because they form a hydrogen bond with these complementary counterparts. So this thickness of the ends which will facilitate the action of the enzyme DNA ligase. So as I told, what is the role played by this restriction enzyme in the process of formation of DNA? So this restriction enzyme which will cut the strand of DNA a little away from the center of the palindrome sites, but between the same two bases on the opposite strands, which will leave the single stranded portion at the end, and these are overhanging structure which is called the sticky ends on the each strand. These are named so because they will form a hydrogen bond with the complementary counterparts. So this stickiness of the ends which will facilitate the action of the enzyme DNA ligase, right? Hope you understood the important concept what we have learned today, right? For in the next class we will study about the further concept of this biotechnology principle and the process.